Okay, welcome everybody for this second days of workshop. So first things, yesterday I sent you a link to download several files. So as you can see here, you have the different presentation and also the advertisements and the requirement for the practical courses. You have a folder with the bioinformatic pipeline. That's the one that we will use this morning. And the statistical analysis, that's the one that you will use this afternoon for the statistic with R. So just a few words about what are inside this folder, because we will work with these files. First, yesterday we got some really interesting question about the primer, design of primer, and especially about the spacer. So I just put here an Excel file. So you have a different couple of primer. This file here is for Foraminifera, and this one here is for V4. So as you can see here, you have the forward primer and the reverse primer. That's the sequence of the primer. Here at the beginning in the forward, here you will have your tag, right? And as you can see, here, the primer is a bit longer, so you have the tag plus the spacer. And as the primer will be a bit far away, it will include some heterogeneity in your sequencing, so it will increase the quality of your overall data set. So this file is just to let you know how I designed the primer in Angers for Foraminifera and also for the V4. Then you have here this first file. It's the pipeline we use first from the FastQ to the demultiplexed files. So this one will be done using Linux. And I comment really every step to let you know what we will do. This is a bash script. So basically you can just run the script directly and get your output. But during this workshop, I will go line by line and explain everything and why we are doing these steps. Then you have this data2 here. It's an R script, and this one we will use to do the clusterization of our reads. And finally, the last one is Vsearch, and we will use Vsearch to assign or ASV to a known reference. And I use Vsearch. Why? Because in the data2 script, you can also assign your ASV, but you don't have any information about the percentage of identity according to the reference database. And depending on the group that you are studying, it's very important. For example, for foram, if you assign a foram to 85%, it's maybe not the right assignment. So for me, I need to have a column with the percentage of identity. Three files here are our scripts, and then we have a row files here. So in the row files, we have a R1 here and a R2. So this is a FastQ that you will obtain from the sequencing platform according to the library. Normally, you will have every time one R1 and one R2 with the name of your library. And this file will contain the pooled samples that you put in the library. So here for the demonstration, I will not work in the whole full FastQ because it's very huge. So I just did a subset of FastQ that contain very less reads. And I recommend you to do that when you are working and on your pipeline to test your pipeline, because you will save a lot of time by doing that. And then once your pipeline is really well set, you can work with your full data set. So a FastQ, yesterday I show you how it works. So you have always 4 lines like this for each reads with the name of your reads, your sequence, a plus, and here this line, the Fred scores. And each of these symbols will correspond to a number, and this number will correspond to the quality of the nucleotides here. And we will use this quality to evaluate the overall quality of your reads and to do some cleaning steps. I also put here the PR2 reference database. So if I open this one, so in a FASTA file, you have only 2 lines and you don't have any information about the quality. So it's really the format you use when you are working with DNA sequencing. And about the title, here you have the number, the gene bank number, what kind of sequence is it, and here's the taxonomy. And with PR2, you have different levels that will be used to assign your ASV. So you will get this information. So it's a great database for many different applications. But if you are working with a special group, with a specific groups, maybe this database is not so well adapted. So you should check also that. But if you are working with the whole diversity of eukaryotes, this database is quite well. And then you also have a tag files here. And this tag files is a FASTA files. And you have each time the name of your sample and the barcode here. So here we have 65 different samples. And these 65 different samples have always a different tag. And we will use that in a step to do the demultiplexing, so to separate the different sample from your FastQ. So that's your raw files. And then you also have some tools. And we will use Vsearch, FastQC, I will show you after, and Flash. And this Cutadapt, in the requirement file, I told you to install them because it requests a lot of libraries like BioPython, or I don't remember which one, but you cannot use this file directly in your script if you didn't install this software previously. Okay, so that's just about the different scripts that we will use. So what I have done a presentation here, and I will show you the different steps. And here you have the shell where we can demonstrate how to run the code. So the first things we will do is a FastQ check. So we will check the quality of our raw data sets. Then we will do the demultiplexing. So we will separate the sample from our raw data using Cutadapt. Then we will trim our primers. So we will remove the primer of our reads. We will just try to merge one sample to check if everything was done properly. Then we will do the clusterization and we will create our ASV using this data2 with R. We will do the assignation with Vsearch. I will provide you some special consideration for forum eDNA and final take-home message. So the first things you should do when you are working with a metabarcoding dataset, when you receive your data from the company, is to check the overall quality of your sequencing. That's very important because you will have the information about the number of reads, the average of the thread score, the presence or absence of unknown nucleotides. Yeah, having an idea about the quality is important because, for example, once we receive a sequencing and we try during several days to do our bioinformatics and it didn't work, and we just realized after several days that the first nucleotides were not sequenced. So it's why our bioinformatics pipeline didn't work. So I just recommend you really to check the quality of your data sets. So there is many software that allow this fast evaluation. Here I propose to use FastQ. So I know that in your computer, maybe it will not work like this. So you should download from the website and try again. But yeah, it's not very important because it's just to visualize how it works. So with this FastQ, it will open a Java Windows like this. You can go, I cannot increase that, I'm sorry. So you go to File, Open, and you will select the FastQ that you want to analyze. You can select several ones and you can open. So the first things you can check is this window here, and you can see that we will work with 100,000 reads. That's the subset. You have also the sequencing length, and for the people that are interested, the percentage of GC. And then if you go to the per base sequence quality, you will have an idea of the overall quality of your data sets. So here, each bar here is the nucleotide. So this one is the first nucleotide, and this one is the end of your reads. So you will always see this kind of patterns with very good quality at the beginning of the sequencing. And the more you sequence, the less good the quality will be, and it will decrease like this at the end. This is a logarithmic scale, meaning the percentage here are really, really high. So you don't have a lot of mistakes in this part, and in this part you increase the number of mistakes. And then this software will provide you different kind of analysis, and you can see also if you have some unknown nucleotide. Here you can see you have all the zero, so you don't have non-sequence base. You have the length distribution. So for Illumina, it makes sense that every reads have exactly the same length. So you can do that for the R1 and also with the R2. Great. Once you have checked the overall quality and you are okay with your file, you can start the bioinformatic pipeline. So to do that, you should be inside the folder that I provide you. And the first things we will do is create some folder, some working folder for the pipeline. So the command to do that is mkdir. And then you have a bunch of folders that will be created. So you can just copy this line and paste the line inside the shell. And then if you look in your folder, you will see that you have created an output folder with different folders here. That's what the command did. The next step we will do is the demultiplexing. So we will use our raw data sets and we will use our tag files, and we will split our raw data set according to the tags files. So to do that, we will use the Cutadapt function, and this Cutadapt function can remove the primers or demultiplex your data set. So about the different parameters we will use. So the first line here is your comment. Then this parameter is 0. So we want no errors in our barcode sequence. We want an overlap of 20. It's not very crucial at that point for this step, but we can set up the overlap. We don't want any insertion or deletion allowed in the tag. And here, this file here is your tag sequence. The first one is first output, the second output, and your input here. That's the general command. And as you can see in the script here, it's a bit more complex because you should provide the path of your different files. So if you look, your tools and your input are in raw file, and you want to add your output in the output folder. So it's why your command here, the one that you should copy paste in the shell, is a bit longer. So you take this command. Okay. So for me, request the executable Cutadapt here that were in the tools. And we are done with this command. If you look on the output you will have here in your presentation, you have a summary here. And it told you that the total read processed is 100%. And he found the adaptators, so the barcode, in 40 0.1% of your reads. That means that it didn't find the barcode in 60% of my reads. So if you look here in the output files, in the demultiplex first step, you will see that here he has created 60 different files that correspond to your barcode. And also you have some unknown R1 and unknown R2, where he didn't find any barcodes. And he didn't find any barcode in these two files. Why? When you do the sequencing, you will not select the side where he sequenced. So he sequenced both in one direction, one half in one direction and one half in another direction. So these two files here, you should reverse complement these two files and then search again your barcode and you will find them. So with Cutadapt, you don't need to do reverse complement and then Cutadapt adapt. You just need to exchange the order of your input here, and it will reverse complement it directly and search for the right barcode. So yes, the second step of the demultiplexing is to search the sequence that have the wrong side and reverse complemented them. So this is the line. It's also a Cutadapt line that will do this job. So you copy paste here this line, and now you will search the barcode in the output files and reverse complemented the things. And in the summary here, he has processed like 60% of the unknown things, and he found adaptators in 46.9% of your file. So if I go to my demultiplex second step here, I have all my different files here, and this is the one, the unknown one, where they didn't find any barcode. It's quite huge, the number here. Why? Because we sequence in this library 96 samples, and I provide to the script only 65 samples. So there is other kind of samples that we will not use in our analysis. So now we have demultiplex 2 times our data set, and we have all the sequence in the right direction. So the second step we need to do is to combine all these two information in a single file. So this is this script here. It's the cat function that allows to copy a file next to another file. So we just take all of this and copy here. Before trimming the primer, we need the files that contain just a list of your sample names. So to do that, we can use your barcode files and remove just the reads sequence. So I will show you the output. And now if I come here, we have 2 new files. That's these 2 commands. And if you open it, you can see it's just a list, text list with the sample name. So you can prepare that by yourself or use these commands. And here we have 2 times exactly the same kind of list, but with, I'm working here with Windows, and it will add each time at the end of each line some hidden characters. And you don't want these hidden characters. So it's why if you are working with Windows, you should remove this hidden character by running this line to remove this R and dollars that Windows will add at the end of your line. That's the kind of differences you will have if you work with Windows or with Linux. Linux will not add these characters, but in Windows, you will have this one. So now in this folder, we have all our samples that are demultiplexed. But they still contain the primers, right? So now the final step for this part of the pipeline is to remove the primer from this individual FastQ. So if I look on this FastQ here, you can see that the beginning of the sequence begin exactly always the same. It's normal because it's a sequence of the primer. And I just copy paste the beginning of this primer and I search this pattern. Here you can see that every reads begin by the primer. And this part that we want to remove. So to do that, we will also use Cutadapt, but this time the function will change because you need to provide the two primer sequencing and say to the program, remove in the R1 the forward primer and remove in the R2 the reverse primer. And what we want also is to remove the sequence that were not trimmed. That's also important. We have other functions that can be interesting. So you can set up a minimum length of your reads. So sometimes it's great to set up this minimum length. It will reduce the noise for the clustering. So basically, it's always this kind of same program. So if you want to use adapt, you should set up this command to your own data set and adapt this command. But the help section of the function is really well done on internet. So now we can come to our script here and do this Cutadapt. So here, as you can see in the step 6, it's a loop. Why? Because we need to go from one sample to another. We have a lot of different samples. So to avoid to set up every couple of samples, you can do a loop like this that will go through all your data in your folder. So you just copy this part and it works. Thanks, Guillaume. And yes, just about the parameters you can use when you trim the primer, you can increase a bit the error tolerance in your primers. Because in the barcode, it's very important to have a 0 because you want that match exactly the barcode. But in the primer, depending on the nucleotide you have, it can a bit change. You don't care too much. So it's why I increase a bit these parameters here. Okay, so now we can check our final trimmed FastQ here. And here you have your list of R1 and R2, and we have removed the primer sequence from this one. So if you check here, you can see that it doesn't start with the same than this one. So it's always great to compare the different outputs and see if you really have trimmed your different primer. See? So now we can confirm that we have done correctly the process of trimming primers. So we are okay with these steps. And then one final check I like to do when I'm doing this step is try to merge just one sample to see if everything went okay and I can merge properly the two R1 and R1 and R2 in a single file. So to do that, I'm using a software that are called Flash that are really doing the work properly. Here you can set up the overlap, the M parameters, the number of threads you want to use in your computer, your first input and your second input. And here it's already done. So what you can see here in the output things is you have 91.6% of your reads that are well combined. That means all your sequences are in the right side and everything works properly, and so your file is correctly trimmed. And if you lose like 10% when you merge your things, it's totally normal. You always lose reads, and 10% it's normally what you are losing when you merge your different files. Okay, so now we are done with the trimming and demultiplexing of our files.
David?
Yeah.
You do the pair and join only for one sample?
It's just basically to check if everything is right. Because for data 2, you need to have as input separate files, not merge files.
Perfect.
But for example, if you are using Swarm or other algorithms, maybe you should merge your R1 and R2 before doing the clustering steps. So it depends the requirement of your files. And just to show you something, this is a different file from the merge algorithm. And if you look on this one, is after the merging where you will the length of your reads. So you will see that here you have the majority of your reads have a length of 137 bare pages at 141. So that's the majority of your read of this one. So it's very homogeneous in the sample data set, and that makes sense because it's a V9 region of the 18S. If you are working with the forums, you will have a very different patterns. And Guillaume told me that with ITS, you also have a huge variation. But with the V4 or the V9, generally, the size of your reads are quite similar. Well, let's start the clustering. So for that, you can open the data2 script, and we will work with R. So this script basically comes from this website, and there is a lot of tutorials that really help you to understand how it works. So first things, if you worked with R, I like to just remove all the objects before starting a new project. So this is this line. So to run the line in R, it's just Control-Enter. So that's what I did just now. Then you have the libraries. You need to load your libraries. So for the data2 library, it's a bit different than the other libraries. So it's why you should follow the tutorial on the internet. That's what we ask you to do before the workshop. And this library, this package, Sekin R, is a great package to build your FastA after everything is done. Then you can save the initial parameters of your R. It's great if you change, for example, the margin. This vector here can help you to go back to the normal. Then this function will help you to get the working directory, and you will have vectors with your path. So basically, here you can see if you just check your path vectors here, that this path will bring you to the folder that we created here with your outputs. So it should bring you to this folder here. Then you will need two files to save the forward and reverse FastQ file names. So you have your path, and then you have the FastQ name that you want to check here. And here it's just a vector with all your different samples there. Here it's again some path of your filtered FastQ. So basically, all this part is just to set up some vectors with path information about your input, FastQ inputs, path information about outputs, and also samples name. It's just to prepare the environments of your studies. And depending on how you write the name of your samples, maybe you will have to change a bit some information in this script here. Then you can plot the quality here of your different files. So here it's not the same than FastQ because it's just an individual vision of the quality of one samples. But you find exactly the same pattern, so a very great quality at the beginning, and then you have a very huge decrease of the quality. So this is for the R1, and you can do the same for R2. And here I just decided to plot two graphs, and as you can see, the quality also decreased a lot here. So now what we want to do before the clustering is to clean or FastQ to have the best quality possible. And then this function, filter and trim, allow you to have different approaches to have the better quality of things. And if you read the tutorial, they told you to cut this part where you see that the quality decrease here. So in our data set, it would make sense to cut here at like 50 and here like at 60. But we will have a huge issues if we do that. Why? Because if you remember, I saw you this histogram and I told you most of the reads have between 136 burpees and 140. So if you could cut here, you will not be able to merge anymore your reads because you will have not so much nucleotides. So what I decided here is to cut at 70 here and cut at 80 here. So I will keep that part, but I need at least 150 burpees to merge correctly my reads. But I decided to increase this parameter here and tell the software, cut all the reads that are below 25 thread scores here. So it's another way to clean your data set. So you should think, okay, I need to clean my data set, but what's the expectation of the length of your reads? And if you cut too much, you will lose some information. So it's not very good. So great. So now I can cut the different files. So here you will have warning, and basically the multi-threading is disabled in Windows. And then if you go back to your output, you will see that you will have a new folder here. And this is all your FastQ that were cleaned. And this is a FastQ that Data2 will use to do the clustering. Here you can see the number of reads that you have before the filtering here, and the reads that you will get after the clustering. And as you can see here, we lose a lot of reads, because the quality at the end was really, really bad. But at the point we did this sequencing, we used ISEQ and not MySeq. So we got much more sequence than expected. So it's fine to lose reads at that point. You can check the percentage of reads you will keep. So for some, you will keep a lot of reads, and for some, you will lose a lot of reads. And if you want to have a look of your plot after filtering steps, you just need to do some new vectors of your path. That's this different step. And then you can again plot the quality. So here you can see the plot before the filtering and after the filtering. So of course, here you will have much more reads, and here you have less reads. But now your FastQ are clean. And it's an important step for your clustering to have some clean reads before doing clustering your reads. And you can do the same for R2, and it gives you quite the same pattern. So now we are done with the preparation of your FastQ. So we can do the clustering of your data set. So the first things with Data2 is to learn the error rates. So for that, we will do two different learning, one for the forward and one for the reverse. Here, it's very fast. Why? Because we used a subset. This kind of steps can take much, much longer depending on the number of reads you will have and the number of samples you get. And then you can plot the error rate to see how he plots according to the different models. So we can apply this error model to our different samples to merge the reads that are similar in ASV. So this is this step where you go through all your samples to use the error model. So you do that for forward, you do that for reverse. And then you can just have some general ID if everything went well, but it's not very informative. And finally, this is a step where you will merge the R1 and the R2. And you can see if you have some ASV, and it seems that it's the case. So you can construct the table that of your community matrix. So all these different functions come from the initial script. I not write myself this script, I just comment a bit more to let you know what's going on. But you should really figure out how does it work. I will not go into the details of each command because it will take too long. But it's just good to give you another view, an idea on how to do this clustering step. So this is just to construct the table now. And here is to plot the distribution of your ASV. And as you can see, it makes sense. We have a lot of ASVs that is 137%. And with this graph, you will be able to see, oh, I cut too much in the filtration step. I don't have anything, or I can cut. Yeah, so you can define better option by looking on this graph. For us, in our case, it's very well, this graph. And now you have a step where you can remove the chimera from your dataset also here. So, of course, with this subset, he will not find any chimera at all. And now what I like to have, and this is something I just write, is to obtain the number of reads you lose at each step of the clustering files. So you just construct a new table here, and you will obtain a table like this. This one is your sample. This is your initial quantity of reads. And then you will see at each step how much reads you will lose. And here, really, we lose a lot of reads when we are doing the filtering because the quality were bad at the end of our sequencing. But then, do the other steps, we don't lose too much reads. And finally, the last column here, the percentage according to the initial content of reads. So we keep just 10%. So of course, with this subset, we don't have much reads, but with the full data set, we still have enough reads to do some statistics. And this step you will see with Guillaume this afternoon. So now in R, you have all your matrices ready, but you should export them in your folder. So first things we want to export is a FASTA files with just your ASV. So we will export two files, one normal and one special for the research that we will use for the assignation. And for this step, we will use the SEC-in package that we load previously. So the function to export your FASTA is write.fasta here. And now if I go to my folder, I have here a new FASTA file that I can open. And I have my ASV, my list of ASVs that I have generated. So the software just sorts the ASV according to the dominant one first. And this number here is the number of reads you find in your dataset. So you find 1,428 reads of this sequence. And in our dataset, we got 62 ASV. So that's the first output you can have. So you can already use these reads in your tree or analysis. But then we can export the specific FASTA for research. If you look what it looks like, I just include the sequence in the title because I will use this information in the script after that. It's just for a technical issue, but it's not very readable anymore. But that's the input that I request for this. But then what is missing now is your community matrix. So for that, you will write another table and you will obtain a CSV with your community matrix. And this one is a CSV, so you can just split. Yeah. And here you have all your sample and the number of reads that you will get per ASV. So that's what you will use for your statistics. And of course, if you want to be sure that everything is well, you can just try to load again these generate files here in your R. And here you will have your T. I name this T. And you can see that you have your matrix ready for your statistic in R also. Now we are almost ready. And the last things we will do today is the assignation, because now we just have a list with all our ASV, but we don't know yet what kind of organism we got. So to do that, we will use a script using Vsearch. So how to assign your things? So you have this tool, it's Vsearch. You will provide the reference database here, and you will tell the software which kind of output do you want. If you look on the help of Vsearch, you will see it's very complete and you can request different kind of output. So for me, I extract some specific output depending on what I want to have as final information. So to do that, you can just run Vsearch, bash, and it work and it will search the specific information, and I need the output to show you what I've done. So now I have a file here, and here I have my different ASV, and I have the number of reads I got for this ASV. I got here the percentage of identity according to the reference database, and here I have the information of what are behind the ASV. So the closer match of this ASV is this reference of gene bank and this taxonomy here. So this is a fungi for this ASV. And if you look, the ASV1, the most dominant one, is also a fungi. So we got a lot of fungi here, some metazoa, some dinoflagellates. This kind of assignation we will discuss more with Enrique. And of course, Guillaume will use a bit this assignation for his statistic. So just to go back on this script here, the important thing is to set up the right output you want from Vsearch. You have the choice between like 10 different outputs, and some outputs are just designed for further analysis. So depending on the analysis you want to do, you can have the right format you want for your analysis. For me, what was really important is the percentage of identity. So maybe right now I'm done with this bioinformatic pipeline. Before concluding, do you have any general question about this? We will discuss in more details this afternoon in the breakout room, but I know that it was really fast, and if you don't know anything about bioinformatics, it's very confusing. And my best advice when you are dealing with this kind of data set is try by yourself. And you should work a lot on that. And once you manage to do several hours on your script, you will be okay to really do this kind of things. It's just an overview, quick, to show you properly how you can do the things. And as you have seen, in like a few hours, we will be able to have all your data ready for the analysis. It's not true, because when you wanted to analyze your own data set, you should adapt all the parameters depending on what you did. And these steps can really take you a lot of time, especially if it's a new group that you investigate. Finish my talk by giving you some special consideration for Foram eDNA. So I use this two primer in the lab here in Angers, and it's the short fragments for forams. But when you sequence that, there is a huge variation of size. Your ASV will range from 60 burpase to 180 burpase. And when you have such differences in the length of your data set, it can be an issue. Why? Because the short read here, the short ASV, will be sequenced more in Illumina and will be amplified more during your PCR. So you will over-represent the short fragments in your data set. So now I need to think how to deal with that, maybe to use corrector factors to reduce this impact. And I know, and sometimes it's true, we don't know yet how to do with that, but in some conferences that are using this approach, you will heard that you have a lot of monothallamides in your samples. So maybe it's true, but monothallamides have a very short sequence. So maybe it's just an artifact of PCR or sequencing. So you should keep in mind. And there is also another issue with Foram, is because for some species, you can have different variants of the same barcodes. So typically for ammonia species, you have different variants that are totally correct, that you can sequence, that correspond to the same species. So that means that you can maybe split a species in several different ASV. So if you split some species but not the other, the weight in your ecological model of the species will be really weak. So maybe we should consider to merge these different variants in a single ASV. But that's something you can test. But if you work at the ASV level, it can be an issue also. And finally, the PR2 database that we use during this demonstration is inaccurate to assign correctly the ASV for Foram. I noticed that it was not really up to date, and there were missing a lot of references for forams. So it's why if you work with a specific groups, it's sometimes really better to have your own database. So as I know that there is a lot of people working with Foram in the audience, that's the things that I'm doing right now in the lab.
So I can summarize. So mainly the question were about the length of the overlap. So why do you trim or not trim the end of the reads to allow your pair-end join at the end of the pipeline? So we can just comment on that, because here we have the special case of the V9, which is relatively short. It was also Illumina, old Illumina CSIC, I think, in that case with 20.
Yeah, it was really the first run we did. And exactly, you're right, Guillaume. It's because it was Illumina, it's only 100 burpase. And if you want... CSIC, yeah. And if you want to have the full length of your V9, you cannot put a 28 burpase for the overlap. So you have to care about these specific parameters. And the message behind that is, depending of your data set, maybe you cannot use by default parameters because it just doesn't work. And in our case, it's why I should set up a very short overlap to keep our ASV. If you increase this parameter, it will just overlap nothing. So this demonstration is just an example, but as you have seen, you cannot apply directly this pipeline to your own data. And there is no pipeline that allow you to do that directly. It's just not possible. Or if there is some pipelines that are a bit more automated, you have no control of the output. and it's just a black box. And basically, you will have some errors that you will not understand. Yeah, your result could be really bad. That's why my advice is just check every script you did and the output and see if everything was okay or not. I see that Valentina asked if we can do the assignation by in Data2. And yes, you can, but I didn't find any option to get the percentage of identity. So it's fine if you want to do the general assignation with the V9 or the V4. But if you want to work with a specific group like TestDataMeB or Forams, you should have the percentage of identity. Because if it's too low, it's certainly not the species of your reference database. So it's why I'm using Vsearch.
Okay, but thank you very much for this answer. But did you compare output? Is it more or less the same because it's based on the same downloaded library, or it's a bit different? Yes. Yeah, for Vsearch and Data2.
I didn't compare the output of taxonomy made by Data2 and by Vsearch.
Okay, but if you know that it's proper way or proper data, you can just use Data2, yeah? It's nothing against this. Okay, cool.
But
thank you.
For me, really, the assignation you will do with Data2 or with Vsearch is just to have an overview of your diversity. But then if you want to use and check really carefully what are living in your things, you should check manually the ASV. And that's what Enrique will explain later, because it will always assign to something, but it's maybe not the real things. So that's what we really be careful when we are doing metabarcoding, because sometimes we lack some reference in our reference database, and we can find some crazy protists that are just not living there, but it's a closer match.
Okay, I see. Thank you very much for this explanation. It's only just sometimes convenient if you don't use Linux or other stuff, just open R and go on. But of course, better to check, double check.
Yeah, and the different categories of PR2, it's very artificial because they split the taxonomy in 7 different ranks, but these ranks are really artificial. There is no biology behind this rank.
Yeah, so, I use also sometimes we use Amphis or something else for package for this. Yeah.
Then the good points about PR2 is also that it's curated, so manually curated, and now it's becoming seriously curated, which was maybe not before. And so yeah, it's Daniel Volo who does that, and I think he does this pretty seriously. And then I think it's a little bit more exhaustive than the other databases like Silva or so for eukaryotes, not for bacteria.
Yeah, I have an interesting question here. I will read this question because it was a direct message. Is there any different result found in the number of ASV and OTUs when you use the same dataset for both analyses? I mean, for example, if you use Data2 algorithms or Swarm, yes, of course, you will have a huge differences in the number of OTUs or ASVs that you will create it. Usually, with Swarm, you will have a much more OTUs than ASV, but apparently there is a new version of Swarm that resolves this issue. But the number of OTU or ASVs that you will get will, of course, depend on the parameters that you will use in your analysis. So, yes, you can find 10,000 ASV or OTUs, or just a few hundred. So it really depends on the threshold, but it doesn't mean a lot of things to oversplit or to just put too many things together.
I have also one question. Before you start your assignments and so on, are you going to check both library like Silva and PR2 and then decide, or you just decide for PR2 directly?
For this example, I use just PR2 directly because it's the most suitable for the V9. But I know that with Silva, there is a lot of people using Silva also. Yeah, sure.
Okay, you mean that for V9 it's better than PR2 now in this stage, yeah? Because for V4 sometimes Silva still, it's everything in dynamic.
Yeah, but this kind of reference database evolve really fast depending on the sequence you will find in GenBank, of course. And the main message in a lot of conferences, we still need taxonomists that provide single cell barcode to increase our reference database. And it seems that it's the case. So it's very, very great.
I would say at the moment, I would advise to use PR2, guys. Yeah. I think it's there are more taxa there, and because this is specialized for eukaryotes, and the people who are working on this are eukaryologists, basically. Whereas PR2, where Silva is like maybe more, yeah, there are a lot of, I mean, if you're working with bacteria, definitely use Silva. There is a question about the chimera check and removal.
I think it's answered, no, but it was just to know at which step it was done.
On which what?
At which step? So when it was done?
After the clustering.
And the pair and join. Because to detect chimera, you need the full biological sequences to be able to detect whether your sequence is built out of two other sequence or not.
Yeah, usually with our meta barcoding dataset, especially with V9 that are really, really short, you will not detect a lot of chimera. I'm sure we have, but we just cannot detect them as chimera. But with V4 region, we have like 1 or 2% of our datasets that are chimera, something like that. But I know that if you have longer sequence, maybe it can be more important to detect that. Okay, I will just
asking about how we manage PCR length bias. So I'm a bit also puzzled out because I'm not sure PCR will induce length bias, more substitution bias.
Or it's not true. It's only on the sequencing that you have this bias. When you did your PCR, the short fragment will be more amplified. Hikel, do you know that? Or
this length bias. Okay, yeah. Yeah, it's on the sequencing. Yeah, sure. When you do equimolar pooling, for example, then if you pull samples produced with different primers and some PCR product created with primers amplifying shorter region, by equimolar pooling, you will have more sequence in a pool of sequence which are shorter than in a pool of sequence which are longer. So that means at the end of the sequencing, you will get more sequence from the samples with more shorter sequence than longer sequence.
Yeah, yeah. Thanks, Guillaume, to clarify this point.
No, maybe and I think Vivien has asked that also, if it was possible, like in your case, with the monothalamides and the other forams, if it was possible to estimate the correct number of each type of foram by correcting with a mathematical, I mean, doing just a mathematical correction.
Yeah, but
I think it would be possible, but we just don't have the function to do that. We still don't know it, but maybe this would be something very interesting.
The best way to resolve this issue, and we are considering to do this kind of experiment, is to extract the DNA from one certain fraction and then split in two. One, you do the morphology, one, you do the eDNA, and you can check really by comparing the results. If you have much more monothalamides, you will see that it's a bias because you don't observe them in this fraction.
So doing mock community
or mock community or all this kind of things. Yeah, no, sure.
I would like to know if there are like a really good page or YouTube channel where I can learn a lot more about the shell unit, because I have a little experience with R, but I never used that shell units because I know that this is like the Linux kind of the CSIC software system.
Okay, so yes, you have many stuff on YouTube and on internet. So basically, we didn't recommend one because it depends also about your mother tongue. Maybe you are more familiar if you find some tutorial in your mother tongue. And also it depends what kind of analysis do you want to do. So basically for Linux, you just need to learn the really basic stuff, how to change your directory, create a new folder and all these kind of things. And for R, it will take a bit more time. And for that, it depends which kind of analysis do you want to do. So for community ecology analysis, we use a lot of vegan. And for that, you can follow some tutorial on how to use and how to build your matrices and how to use this kind of things, yes. So you should start one tutorial, and if you think it's not useful for you, change and find the good one that will help you. So you should really try several ones, and once you have a good one where you learn really something, you can keep this one.
Okay, thanks. So I need to focus on R more because there's like the more general software in the research group we use.
R, it's more for the statistical part, and Linux is more for the bioinformatic part. And this morning I presented the clustering in R because this Data2 software were designed for R, basically. But normally you do all your bioinformatics using Linux. And finally, my take-home message for this presentation, I know it's very hard to follow this kind of bioinformatics course. And even if you work in bioinformatics, it's sometimes difficult. It's too fast, I know. But it's just to give you an overview on how to do the things. And now if you are really interested, you should do and work with your own data sets. That's the best way to learn bioinformatics and also statistics. So my take-home message for my part is you should use the most up-to-date version of all the different algorithms that you will use. For example, now I know that the v3 of Swarm is just out, so you should switch to this version because the most up-to-date will resolve some issue of the previous version. So it's always great to update your pipeline. And this field of research evolves really fast. So to be updated, it's sometimes very challenging. Second things, you should check the results of every function in order to be sure that everything was done properly. When you run a command in your shell, you should check the output, like I show you in the text file and show you the, yes, look, we remove the primer, it's well, and you check visually that everything is right. Because in some script, it will not tell you that something went wrong. So by checking manually the output, you will avoid to lose some time. And the last thing is to try different approaches to study your data set. You should obtain more or less the same output, meaning if you use Data2 or Swarm, the dominant species should remain the same. If you have a complete different pictures, you have a problem with your data set. But all the tests that I see with bioinformatics, you will obtain more or less now the same kind of output or the same kind of message. Of course, there is small differences depending on your reference database you will use, depending on some parameters you will provide to the algorithm. But still, the overall value of your data set should remain the same. And when you have more experience with this bioinformatics and statistics, you can set up more precisely all these different parameters and improve your signal and the quality of your results. Thank you very much.